Today we're going to talk about proteins. And proteins have many different functions. Proteins are going to be used to make enzymes, which speed up chemical reactions. Proteins are also going to be used to make structural elements, such as the cytoskeleton inside the cell. And proteins can also be used to make messengers, like hormones, such as insulin. Proteins are going to be made of monomers called amino acids. And there's going to be 20 different amino acids. And by putting those amino acids together in different combinations, we can make limitless numbers of proteins. Most of the proteins in your body are several hundred to maybe even 1,000 amino acids long. Some proteins can be over 1,000,000 amino acids long. So let's go ahead and draw an amino acid. We're going to start by drawing a carbon atom. That's going to be the center of our amino acid. Carbon forms 4 bonds, 1, 2, 3, 4. Over here, I'm going to draw a carboxyl group. C double bond O, OH. That's a carboxyl group. It's a type of functional group. Over here, we're going to draw another functional group called an amino group. It consists of a nitrogen connected to 2 hydrogen atoms. That's an amino group. Again, it's going to be a very polar functional group. And both of these functional groups are going to be sites of chemical reactions. Up here, I'm going to draw a hydrogen atom, and then down here is referred to as an R group. There are 20 different R groups, combinations of atoms. And by putting different R groups on here, different combinations of atoms, I can create 20 different amino acids. Now this part of an amino acid right here is referred to as the backbone. All amino acids have the same backbone in common. Down here is referred to as the R group. And again, by substituting in different R groups, we create different amino acids. So let's go ahead and connect up some amino acids. So there's one amino acid. Now I'm going to draw another amino acid over here. So there's our carbon, 4 bonds, a carboxyl group, an amino group, a hydrogen, and an R group. Now I want to connect together these 2 amino acids. And once again, you can see I have a hydrogen. We're going to break this chemical bond between this nitrogen and hydrogen. And we're going to break the chemical bond between the carbon and the oxygen. I'm then going to take this hydrogen and this hydroxyl group and remove a water molecule. At that point, this nitrogen is then going to form a chemical bond with this carbon atom right there. So let's go ahead and get rid of this water molecule here. Clean this up a little bit. And I'm going to draw the connection between nitrogen and carbon. Now it's not really that long, but for purposes of explanation, let's make it that long. Now this is a covalent bond here. This is a covalent bond, that's a covalent bond, 2 covalent bonds. But what makes this covalent bond special is its location. It actually connects together the 2 amino acids. And because of that, we're going to give it a special name. It's going to be called a peptide bond. Now it's important here that this chemical reaction only occurs between an amino group of 1 amino acid and a carboxyl group of another amino acid. It doesn't happen carboxyl group to carboxyl group or amino group to amino group. It has to be an amino group to a carboxyl group. And that creates a dipeptide, 2 amino acids connected to each other. Now if we want to connect the 3rd amino acid, we draw another amino acid and again connect it up via a dehydration reaction. And if I want to break apart these amino acids, if I want to break this protein apart, I'll add the water back through a hydrolysis reaction. And that is how we're going to put together amino acids to make a protein.